After studying this module, the readers shall be able to understand the forensic DNA typing techniques developed so far since 1984, the steps involved in techniques and their limitations, the interpretation of the DNA typing results. Forensic DNA typing, also referred to as DNA fingerprinting or DNA analysis or DNA profiling, is the most significant advancement for use in justice administration in civil and criminal cases. The discovery of highly polymorphic regions in human DNA by Wehrman and White in 1980 laid the foundation for the discovery of individual specific banding pattern observed after restriction fragment length polymorphism analysis of repeated DNA sequences by Professor Sir Alec Jeffrey at the University of Leicester. He found certain regions of DNA containing DNA sequences which were repeated over and over again next to each other. He also observed the number of repeated sections present in a sample could differ from individual to individual. These repeat regions were known as VNTRs or variable number of tandem repeats. The technique used by him to examine the VNTRs was called restriction fragment length polymorphism or RFLP, as it involved use of restriction enzymes for cutting the regions of DNA surrounding the VNTRs. The breakthrough in DNA fingerprinting focused the attention of forensic scientists of Home Office Forensic Science Service UK, and the procedure for extraction of DNA from dried strains of human body fluids were developed. Subsequently, commercial introduction of the PCR process in 1987 accelerated the pace and widened the scope of application of DNA technology in the forensic field. Several commercial laboratories like Cellmark Diagnostics in UK, Lifecourts Corporation and Cetus Corporations of USA played significant role in developing and standardizing various techniques and protocols. After extraction of DNA from the target tissues, its purification and assessment of its integrity, earlier RFLP method was used for DNA typing, which was subsequently replaced by PCR-based methods. RFLP method encompasses fragmenting the DNA strands into desired lengths using restriction enzymes, separating the DNA fragments through the gel medium according to their size or molecular weights, transferring the fragments onto the solid supports, nylon or cellulose membranes, hybridizing the specific DNA fragments with complementary DNA pieces or probes tagged with radioactive or non-radioactive material, and taking the images or impressions of the desired fragments. PCR-based methods involve amplification of a small or specific portion of DNA or its loci for further manipulation for desired goal. RFLP typing using multi-locus MLP and single locus SLP probes. Sets of base sequences repeated numerous times are present within the human genome. These repeated sequences are called mini-satellites, which show a very high degree of allelic variation in the number of repeat units and accordingly in their length. Hence, they form the basis of differentiating or identifying individuals based on this length polymorphism at molecular level by using either multi-locus or single locus probes. Use of only one multi-locus probe can provide sufficient numbers of variable bands which establish positive identity of an individual. Thus, it is a single powerful test for positive matching of body tissues and determination of parentage. Probability of obtaining identical patterns of bands from the DNA of two individuals using multi-locus probe is of the order of 1 in 10 to the power 14 to 10 to the power 30, which is more than 5 into 10 to the power 9, the total world population. Thus, the DNA prints obtained from multi-locus probes are highly unique. For detecting variations at specific mini-satellite loci, single locus probe or mixture of two or more probes were used. A single locus probe reveals a pattern of up to 2 bands, whereas mixture of several probes reveals more than 2 bands. These probes are highly sensitive and hence can be used for small and even partially degraded samples of DNA. These are useful in identification of mixed samples as in cases of multiple rapes. The RFLP DNA testing has 4 basic steps. First, restriction digestion. After isolation of DNA from any biological sample, the specific quantity of DNA sample is treated with restriction endonucleases. For example, hint 3, PST1, etc. This step is called restriction digestion of the target DNA sample. The endonucleases are capable of recognizing and cutting the DNA at specific sites along its sequence, resulting into a reproducible set of restriction fragments of various lengths. The restriction enzyme cut the DNA into thousands of small fragments of nearly all possible sizes. Second, separation of restriction fragments. Fragments of DNA are separated according to sizes on agarose gel under the influence of electric current, that is electrophoresis, wherein the gel acts like a sieve through which small DNA fragments move through the pores of gel rapidly as compared to larger fragments. Third, Southern blotting. The separated fragments are visualized by Southern blotting, where membranes are transferred from the gel onto the nylon membrane. Fourth, hybridization. The DNA fragment fixed onto nylon membrane is hybridized with either multi-locus probe or single locus probe. In single locus probe, each probe is specific for a single trait or single locus in the genome. These probes are DNA sequences labeled with radioactive or chemiluminescent substances. Multi-locus probes consists of tandem repeats containing a mini-satellite core sequence capable of simultaneously detecting a number of highly polymorphic loci having same core sequence and thus generating individual specific DNA fingerprints. If radioactively labeled probe is used, The nylon membrane or blot is laid onto a X-ray film. Bands are formed on the X-ray film on the site of radioactive substance present on the blot. This X-ray film is called autorad. Alternatively, the bands are visualized using chemiluminescent dyes. First application of DNA fingerprinting, in which two multilocus probes were used to analyze samples for immigration case. In this case, sample of mother, her disputed male child, 3 undisputed children and 1 unrelated individual were analyzed by Jeffrey. PCR based typing, reverse slot blot, AMP, FLP, STR typing, mtDNA sequencing and SNP typing. Some VNTR loci have relatively short size alleles. These loci can be PCR amplified. Locus D1S80 was used in forensic setup for AMP FLP analysis. The fragments in this locus are in range of 14 to 42 repeats unit, that is 16 base pair per unit. The amplified fragments were separated according to size using polyacrylamide gel electrophoresis and detected using silver stain. The discrete alleles are compared directly with the allelic ladder. The AMP FLP technique requires less DNA than RFLP method and worked well for degraded samples. This locus can be analyzed in complex fashion and amylogenin locus for gender typing. Short tandem repeat or STR, a subcategory of VNTR, is a region of human DNA containing 2 to 7 base pairs repeat per unit. These STRs are also called microsatellite or simple sequence repeats. More than 10⁵ STRs exist in human genome, out of which some STRs have been characterized specifically for forensic DNA profiling. Earlier, the STR profiling was carried out using polyacrylamide gel electrophoresis of the PCR product and staining the gel with silver stain. Figure 6 shows the STR typing at 9 loci, 3 on each plate, where sample number 1, 2, 3 and 4 display the allele, which is given number against the allele in the ladder run along with the sample in the gel. Presently, STR profiling is being done using multiplex PCR system, in which multiple fluorescent dyes are used to detect the amplified product using capillary electrophoresis in automatic DNA sequencer. Sequence polymorphism at DQA1 and 5 other polymorphic loci on human DNA were analyzed based on the phenomena of complementary base pairing. Under appropriate condition, only those single strands which match exactly will hybridize. If only one or a few bases are different, the two strands will fail to attach to each other. Commercially available nylon strips to which DNA probes are attached are treated with PCR products. Finding complementary sequence, the target DNA fragments in PCR products bind to the strip at the location of the probe. The primers used in the reaction are labeled with biological tag called biotin. on reacting with protein streptavidin or horseradish peroxidase and TMB with hydrogen peroxide, blue color dots appear at the site of binding of DNA sequence with probe. Pattern of dots correspond to alleles present in the sample. The results are recorded and saved by photography. Mitochondrial DNA sequencing is carried out when the samples are old or badly degraded, often bones, teeth and hair, or in cases where amount of nuclear DNA is very small for STR profiling. The Sanger method, developed in 1977, is still widely used for mitochondrial DNA sequencing, in which each DNA strand is sequenced in separate reactions with a single primer, either forward or reverse. 4 different colored fluorescent dyes are attached to the 4 different ddNTPs. These are similar dyes to those used for STR detection. DNA sequencing of mitochondrial DNA is carried out in the following steps. First, PCR amplification of the entire control region or a portion of it with various primer sets. Second, removal of remaining dNTPs and primers from PCR through spin filtration or enzymatic digestion. Third, determination of PCR quantity. Fourth, performance of DNA sequencing reaction to incorporate fluorescent ddNTPs, with each reaction containing a different primer to dictate which strand is sequenced. Fifth, removal of unincorporated fluorescent dyes terminators from the completed sequencing reaction. Sixth step, dilution of purified sequencing reaction products in formamide. 7. Separation through capillary electrophoresis instrument. 8. Sequence analysis of each reaction is then performed. Single nucleotide polymorphism or SNPs are used for human identification, paternity testing, hair color and eye color identification in forensic setup. Over the years, a number of methods have been developed for SNP analysis, such as measurement of fluorescence, luminescence and molecular mass. Most assays are carried out in solutions or on solid matrix support, such as glass slide, chip or bead. Autosomal SNPs can be used for many types of forensic testing, including analysis of degraded samples. SNP loci on Y chromosomes are also potential markers for paternity testing because of low mutation rate. Now, let us summarize what we have learned in this module. DNA profiling was developed due to 2 independent breakthroughs in molecular biology at the same time in UK and US, where polymerase chain reaction was developed by Carey Mullis of Cetus Corporation, US, and individual specific bending patterns were observed after restriction fragment length polymorphism analysis of repeated DNA sequences by Professor Sir Alec Jeffrey at the University of Leicester, UK. In RFLP technique, multi-locus and single locus probes are used. This technique is capable of generating highly individual specific DNA profiles. Since in this method relatively large amounts of DNA are required, it is less vulnerable to contamination by extraneous sources. However, it suffers from several shortcomings like the limited sensitivity of detection, due to which it is difficult or impossible to obtain meaningful profiles from trace biological evidence or from samples that have significantly compromised due to aging, etc. In addition, the interpretation of VNTR profiles also difficult sometimes due to artifacts of the procedure such as partial HAE3 digestion, band shifting and incomplete profiles due to faint hybridization of low molecular weight alleles or degradation of high molecular weight alleles. RFLP typing is quite time consuming, laborious and difficult to automate. Limitations of RFLP typing have been overcome through the development of PCR based DNA typing systems.